Hey everyone, thanks for having me speak at this session. As Kai mentioned, I'm going to be talking to you about single cell sequencing and some spatial transcriptomics technology. And I'm going to be focusing this talk mainly around development. And development is really quite a fascinating thing. So I always show this kind of video at the beginning of these talks, just to kind of illustrate how we start from a single cell, but that single cell quickly kind of divides and develops into what very quickly resembles an organism. Here we're looking at a salamander, and this movie is kind of sped up in terms of its time component, but you can clearly see over its course how that single cell kind of divided, and finally took this final form. And many scientists like to abstract this process as kind of this single cell sitting on a landscape. So here we have this cell represented as a ball. So that cell is kind of this totipotent stem cell where sperm and egg come together. And this cell can give rise to all the different tissues in our body. And the way we think about this landscape is that each of these different kinds of regions represents a different set of tissues that this stem cell can go into. For example, if this ball kind of rolls into this area, it'll form muscle cells, cardiomyocytes, connective tissue, and blood. And as I play this movie forward and you see these kind of spheres roll onto this landscape, they start to populate all these different areas in terms of cell types. And a lot of the single cell data that we collect actually ends up taking these kinds of representations. So I'm going to pause this video and kind of just take a look at it at a single cell to kind of explain this. So here we're looking at a single cell transcriptome. And what I mean by that is we've effectively counted the number of molecules present for about 20,000 genes in a cell. And when we're looking at this plot, the way to interpret it is that a cell's position on the scatter plot actually denotes similarity in gene expression. So if you have two cells that are near each other, they're going to be very similar. And two cells that are kind of far from each other are going to be more dissimilar. And from this kind of data, we also make inferences. So for example, if we were to see these gray cells, green cells, and yellow cells, we may make the inference that the gray cells are early in development, they're stem cells, and that they differentiate into these two different kinds of cell types, the green cell type and the yellow cell type. And because we've collected this gene expression data for each of these cells, we can also start to infer what kinds of programs are being turned on in each of these cells. For example, if we look down this branch of green cells, we may see that all of these cells start to express gene A, and these cells on the right start to express gene B. And in this way, we can make hypotheses or test hypotheses as to how this kind of cell differentiation process is happening. And these kinds of single cell atlases, as we call them, have been collected from many different organisms. So here I'm going to show you three different data sets, one for the embryonic nematode, C. elegans, embryonic zebrafish, and one for mouse gastrulation. And these kinds of data sets have been collected for many different organisms, and they span many different stages of developmental time, and they're even now being collected for disease. But one thing to note in each of these images is that I've colored the plots going from red in early development to blue in later development. And what you'll notice is that there's a kind of a smooth progression between cells that are early in development to later in development. And this really kind of recapitulates or represents that earlier kind of schematic I showed you of those spheres kind of cascading over the landscape. But kind of the use of the word atlas actually kind of denotes that these are maps of some kind. And a question that I want to ask or kind of put forth is, are these single cell atlases really maps? So when I think of a map, I think of this. So here we're looking at kind of a Google Maps view of Paris. And maps are pretty useful because they actually help us navigate between different points. So for example, like let's say that I was at this restaurant near the Eiffel Tower and I wanted to get to the Louvre. I could put this information into Google Maps and I would effectively get out a set of directions, take these three turns and you'll end up at the Louvre. these maps, at least in terms of Google Maps, are static because they're fixed representations, but they can convey rich and dynamic data. So for example, if there's an accident here, maybe Google Maps can help us reroute and find a different way to get where we're trying to go. So one of the kind of key questions that I've been thinking of, and many others in the field, is can we build comparable biological maps? One, they need to be navigable. We need to get directions from point A to point B. They need to be able to outline a set of paths for travel. So in the maps example, these are the roads, sidewalks, bike lanes, and that really depends on the mode of transportation. And finally, we'd love to have a static representation of this kind of biological system, in the sense that as we add more data, that map becomes richer, but its kind of underlying form is unchanged. And when I think of this third point, I think of the human genome's kind of linear sequence and how the ENCODE consortium has been able to layer on kind of multiple modalities and layers onto that linear sequence. Having something comparable for development or tissues would be kind of ideal. And to kind of take this analogy one step further, the single cell atlases that we've been generating are really analogous to street views. So here I'm showing you 3 different clusters of cells. And in the maps analogy, this is really kind of analogous to kind of 3 different sets of street views that look very similar to each other. So for example, the set of street views over here on the left, each has a door. So that's why they're kind of being clustered together. There's a park down here, and on the right, maybe these are all beige buildings. So this really doesn't help you get between these 2 places. And you can start to see like how our kind of using similarity in transcriptome starts to break down in terms of using them as maps. And as I mentioned, what we really want to get towards is kind of taking this kind of rich molecular resolution, which we have on the left, which are these whole transcriptome measurements, and starting to combine them with kind of some time-resolved tracking of a cell's 3D coordinates. So this video on the right is real data of a mouse gastrulating. It's a beautiful video that came from Katie Dole and in the Keller lab. And what you can see is each of these cells are being imaged over time, and this organism is effectively developing. But unfortunately, we don't actually have any molecular data. We don't know which genes are being expressed by these cells in each of these positions. So there's been an explosion of different methods in terms of collecting this kind of data, meaning both the combination of this molecular resolution or molecular contrast along with kind of imaging or position. On the far left here, we have light sheet, which is the data I just showed you from McDowell et al. It's diffraction limited, but you do get the single cell resolution. Unfortunately, though, you can only detect a very low number of genes. But one of the best parts about this is that it's live imaging, which you'll notice is not a feature of any other of our technologies. Other such technologies include multiplexed in situ hybridization. There's been a number of papers here and some exciting new companies and technologies that have formed around this concept. Spatial RNA-seq is another concept where you can effectively capture RNA molecules on barcoded beads or spots. And I'll talk further about this later in the presentation. And finally, I'm going to spend some time today talking about some of my work, kind of doing collecting single cell data, but also collecting fiduciary for spatial component as a part of that data. And this method is called SkySpace. And to kind of just briefly walk you through how this works, what we do is first we have a glass slide with a layer of gel as a backing. And what we've done is we've kind of spotted different combinations of oligonucleotides such that each of these spots has a different combination of oligos. This kind of layer here is about 22 millimeters by 22 millimeters and contains about 7,000 spots. And here you can see some of the kind of measurements between the spots here on the right. So to use this slide is very simple. What we basically do is we take our gel-coated slide we take our embryo section and we effectively sandwich them together. And what we're doing is we're effectively stamping our spatial positions from this one kind of coded slide onto our cells in our embryo. Then when we dissociate our cells, we can effectively perform single-cell RNA sequencing to measure both the RNA contents of each cell, along with these oligos, which will kind of give us a cell's relative location in that grid. So for example, that cell, for which we saw these two oligos may correspond to this position here in the grid. This is kind of at this point, this is like a relative position. And we kind of lost some of that measurement from just visualizing the embryo itself. So to kind of conserve that information, we can also mark some of these spots with a fluorescent molecule, cyber green. And so then when we transfer these oligos onto the embryo, these fluorescent spots also get transferred. And effectively, these fiduciary points help us align the sequences to this image itself. So using this method here on the right, I just want to kind of quickly contrast this method to other methods of collecting spatial transcriptomics data. So on the left, this is kind of a more conventional approach where you're laying a tissue here in this kind of like liver-looking structure onto these beads. And what's happening here is that you're actually collecting the RNA from each of these cells onto these beads. So the resolution is really limited by the beads itself. Whereas here on the right, because you're kind of moving the, stamping these kind of oligos onto the cells, you still have single cell resolution, but your spatial resolution is now confined by how kind of finely you can put these kinds of spots down. One thing that's notable about this kind of data is that it does look like single cell data, and it is single cell data. So over here on the left, I'm showing you one of these scatter plot embeddings again, where each of these clusters is a different cell type. And what you can see here for about 120,000 cells is that we recover many of the different kinds of tissues present in the mouse embryo when we perform this technique. And not only do we have that kind of information, we can break out each cell and ask which slide and which kind of region of each embryo do these cells come from. On top of that, we have that molecular contrast that I mentioned before. So for each of these cells, we can also kind of see which set of genes they're expressing in addition to where they're coming from in the embryo. So on the left, I'm showing you kind of the data from one of the slides, and these cells in green are expressing the dopamine transporter, SLC6A3. And on the right is a matched kind of in situ hybridization experiment from that same brain region. And one thing to kind of that's notable and you could kind of see on the last slide is that we're only collecting about 2 to 5% of the cells in a tissue. So there's a lot of room for improvement here. So when we compare kind of how many cells we think there are via imaging versus the number of sequence transcriptomes, we're only capturing a fraction of what's there. But even with that fraction, we can do some pretty amazing things. So for example, I'm going to kind of focus in on this set of neural cell lineages here and kind of walk you through, again, why kind of having this spatial information is so important. So if we kind of break this area down and kind of re-embed it, it takes this structure where you have these progenitors here, which are radial glia, and you have 3 different types of neurons. So if we were to kind of follow that same set of inference or logic that we had before, we would assume that these radial glia are actually differentiating to these different neurons. But because we have this spatial information also, we can actually go back and ask, where are each of these cells sitting in the mouse brain? So to do this, we can manually segment the brain regions using the Allen Brain Atlas, and we can localize each of these cells anatomically. And when we do that, we see that each of these 3 prongs here actually corresponds to a different region of the brain. So each of these kinds of differentiation trajectories is happening in an anatomically distinct region. We can also kind of derive a measure of kind of progression from radial glia all the way to neurons in each of these kinds of anatomically distinct regions. And we can calculate kind of a pseudo time or a normalized kind of distance along each of these axes for each of these cells. And when we do that, and when we visualize the pseudotime spatially, it becomes pretty apparent that what we're kind of witnessing here in terms of this smooth trajectory in transcriptomes is actually a spatial phenomenon itself. In this case, it's this process of radial migration where these precursor radial glia are born around the ventricles. And as they differentiate, they move towards the periphery of the brain. And you can see that really clearly here, where the V here is the ventricle, cells in blue are early in pseudotime, and so as they move towards these yellow cells, they're differentiating and migrating. We can also look at the set of genes expressed during this trajectory to kind of back that up. They're mainly genes involved in cell division and cell cycle initially, and then eventually lead to projection, morphogenesis, cell migration, etc. So then one of the kind of visions I laid out earlier was that can this spatial data actually start to resolve spatial position in single cell data sets? If we're going to start to integrate these things, we're going to have to kind of transfer labels from one data set to another. And to do this, we took this paper published by Lamano et al. In 2021, which has a molecular architecture of the developing mouse brain. So this is an atlas of brain development in the mouse. And what we did is we integrated these two data sets. So what we can do is effectively look across two different data sets and find a gene expression which is similar amongst them and calculate a new embedding that effectively brings these two data sets together. And by doing that, we can kind of transfer labels from one data set to another. And there's been a lot of different algorithms and a lot of work done in this area. This is not even close to a comprehensive list of papers, but this has been a very hot area in the single cell genomics world. And so when we do this with the two data sets, we see that they overlap nicely. On the right is the sky space data that has a spatial kind of component, and on the left is the single cell data from Lamano et al. When we kind of transfer the dissection labels from their data set to ours, they largely make sense. You know, when we look at cells in the dorsal forebrain or ventral forebrain, they kind of match up to the anatomical location we'd expect in our data set. And what's kind of, you can take this one step further and effectively look at the set of clusters that they've defined in their data set and ask how they kind of co-localize with the spatial coordinates in our data set. And what we find is, in the brain at least, about 60% of these kind of transcriptomically derived clusters co-localize spatially. And what this effectively means is that when we look at these kind of embeddings, they really contain a convolution of many different things, one of which can be spatial position very strongly. So I mentioned that this area is kind of rapidly moving and is an area for rapid innovation. And I think I would be remiss without kind of mentioning this new technology that's come out in the last year and is totally blowing people away. So this was published in Cell, and I think there were a number of papers, I think 6 to 8 papers that came out from this group. And this is the stereo-seq technology from the Beijing Genome Institute. I'm sorry. One second. Yeah, so what they do in this technology is effectively they have these barcoded spots, which are kind of at half a micrometer resolution, kind of arrayed across a different area. And they can effectively sequence each of these barcoded spots before kind of capturing RNA in situ on them and then sequencing them again. So on the right, you're looking at a figure from their paper. Now, instead of this kind of lower resolution that I was showing you earlier, you have a much higher resolution where you can actually segment the cells from the image that you've taken and kind of overlap them with these nanoballs that are within each of them. So here they're kind of highlighting different epithelial cell types located in different parts of the developing mouse. So for example, the AT2 cells in the lung kind of nicely illuminate the alveoli, which are present in the lung, or these follicular epithelium, which will eventually give rise to whiskers in the mouth. So this is a kind of really amazing and cool data to see. So as this kind of field goes forward, it's going to be imperative, I think, that we move from acquiring kind of these 2D data sets that I've shown here to more 3D data sets, continue kind of integrating with other imaging data sets. The final goal of this kind of thing, I think, should be kind of making a molecular resolved movie or model of a biological process. And I think, these technologies in principle can be applied to a range of biological systems, from development, as I showed you here, to kind of disease and pathology. And as the drops in sequencing kind of happen and sample prep, hopefully these technologies can penetrate kind of medical markets and other kinds of markets where they can gain kind of wider spread adoption. So in conclusion, I've kind of told you these single cell genomics are being used to study development. So our technology, SkySpace, is able to measure both single cell transcriptomes and spatial position by effectively stamping these positional oligos onto cells. And these methods can now be used to integrate kind of existing data sets to recover how spatial position kind of is important in some biological process. And finally, I think there's a number of groups that I would recommend kind of following in this area. And that includes like Xiaowei Zhuang's group, who's developed MERFISH, and they have the new kind of VizGen instrument as their kind of commercial platform. StereoSeq, which is that Beijing Genome Institute technology, PixelSeq from Liang Kai-gu here at UW, StarMap from Xiao Wang, SlideSeq and dbitSeq. So write these down. These are great people to kind of like follow if you're interested in the development of this field. And if you want to kind of learn more about the work that I showed you today, you can check out our paper that was published in Science in 2021. We had this artist kind of do a really cool kind of rendition of the developing mouse embryo with each of the different cells kind of having their own personality. You can find this on the internet on my Twitter if you're interested. And with that, I'd like to thank sources of funding, the Shinduri Lab and the Trapno Lab, and my co-authors, Mary Reiger and Kelly Stevens.